So, I would like to start this topic with a demonstration. I have requested Ratin to help me out with the demonstration. I needed to start it out with the demonstration rather than with the theory, because you see the process and then understand how it is seen in real life, and we will work back into the theory part of it and why it is a very fundamental topic in motor control, that is the control of all motions within the body. So, what I am about to demonstrate here is something which some of you who have gone to a neurologist would be familiar. So, this is something called as the demonstration of a reflex, and anybody from a MBBS background would realize how difficult it is to show this to an examiner in particular. So, what is being shown is a tap, and that is causing the limb to move. A similar kind of movement, not very obvious, is the supinator reflex, and this is, yeah, you can see that is the triceps reflex. So, for better viewing and for continuity, I think I will demonstrate on the opposite side. So, this is the biceps. Yeah. So, Ratin, I want you to do one thing. You try to hold your hand stiff. Yes. Note the difference. It is just almost disappeared. Okay. So, we will do the same thing with in true scientific methodology, repeatability. So, Ratin is focused on the triceps. So, I am not able to get the triceps as well as it is on the opposite side. So, these are called reflexes. These are part of the clinical evaluation of patients and people when they check for their neurological status. But for the purpose of our discussion, it is important that you understand that this is universal. See, I have demonstrated it on Ratin. It would be very easy for him to demonstrate on me and vice versa, and for any given person in the population. There are a couple of these reflexes, and I showed it to you on both sides of the body because it's a universal phenomenon. So, starting from that, I think we will go on to the theory of how these reflexes form the fundamental mechanism of control and movement within the nervous system. So, the topic of discussion is something called as reflexes. So, reflex is a medical term in which you find stimulus which produces a specific response. Now, the response is specific, that is important. It is not just any kind of response. So, there is a specific stimulus, and that way produces a specific response. In the video, I have demonstrated something called biceps supinator and triceps, both left and right, and I showed something about how it is universal. So, there are a couple of such reflexes all throughout the body, and they form a class of reflexes called monosynaptic reflexes. These, excepting of course this is polysynaptic. So, we will see what actually happened during the process, and then how it is very important that we understand the process. So, going back to our very old discussions, we understand that there are muscle cells which can contract, and so this is general notation for a muscle cell. So, how a muscle cell acts is because it has got a synapse, and the synapse has got a axon which comes all the way from the spinal cord. So, the cell body of the synapse is in the spinal cord. Now, this is one part of the story. So, what actually happened during the process is muscle cells are attached to the bones, okay, and this is a representation of a bone, and when you tap that hammer, so this is through tendons. So, this is a tendon, muscle, okay, muscle cell, tendon and bone. So, when you tap, when you use that hammer and tap, what happens is there is a sudden increase in the length of this muscle cell. Now, what this results is, it results in the activation of something called as a muscle spindle. Now, muscle spindles are sensor systems. I call the term systems instead of a cell, because we will subsequently try to understand what its complete functions are. So, the muscle spindles are located in muscle fibers, and they are these kind of spindly structures which is present within the muscle cell, and they are parallel. So, what I mean by parallel is, when there is a change in length of the muscle cell, there is a increase in length of the muscle in the spindle also. So, muscle spindle also gets increased. Now, the muscle spindle is not so simple. There are fibers which come along the surface here, and these fibers in turn go all the way to the spinal cord through the sensory part of the nerve, but the axonal part of the neuron would go and synapse onto this muscle cell. And the more interesting part is, it gives a positive stimulation. So, let us analyze what has happened. So, we have a basic resting muscle spindle, sudden tap causing an increase in the length of the muscle. That length causes an increase in the length of the muscle spindle, that in turn activates these fibers. So, basically these contain sodium receptors which are sensitive to stretch. So, there is an increase in length, this cell gets activated, and by default it is synapsing to the muscle cell of the same muscle, the neuron supplying the same muscle, there is a positive activation, and what actually happens is it stimulates. Now, what does stimulation result in? Stimulation results in decrease of the muscle cell. So, what happens is, contraction of the muscle cell. What happens to the muscle spindle? Muscle spindle also reduces its size, and what happens to the neuronal stimulation? Nothing happens to the muscle cell. So, you have negative output here, and that in turn produces negative. Okay. So, this is the theoretical demonstration. So, we will reiterate the whole stuff again. Sudden increase in length of the muscle causes increase in length of the sensor. It is a stretch sensor. The sensor gives an output to a neuron. The neuron gives an output to the commanding neuron of the same muscle cell, and that reduces the size of the muscle cell, and then it contracts. Once the length comes down, the output from the receptor sensor comes down. The neuron which is supplying the sensor reduces its activity, and the muscle cell activation comes down because the neuron coming to the muscle cell reduces its activity. So, that is what we actually saw. The key step here is this. So, we see that in the entire exercise there is only one synapse. So, the single synapse is what makes it called as a monosynaptic reflex. Yeah, so far so good. So, it looks very interesting, it looks very novel, but what's so fundamental about it? There is nothing fundamental about this, right? So, what I have told you is only part of the story. So, part of the remaining part of the story is what makes this system very interesting. So, muscle spindle is not just about that. There is still one more entity which needs to be considered. So, muscle spindle, I drew it like this. Spindle. So, spindle is shaped like this. Apart from having this, there are muscle fibers in this part of the spindle. That is what makes it interesting. So, you have something called as intrafusal fibers. So, these are intrafusal fibers and the others are extrafusal fibers. So, extrafusal fibers are outside of the spindle. So, we are looking at it from the spindle point of view. So, this is the larger muscle cell. And these have synapse over here. These have synapses over here, and then there is an entire sensor system sitting over here. And you have two muscle cells which can be activated independent of each other. So, this is called 1 beta and 1 alpha. They have rates of transmission which are very different. Now, let us look at the now we have set up the discussion. We have set up the baseline, and we will see how things work from here. So, what actually happens if we send a stimulation to 1 beta? So, 1 alpha, 1 beta, then spindle muscle cell, or maybe I think I should make it a little more detailed and we look from top down. So, anterior cell neuron, this is in the spinal cord. This goes to skeletal muscle cell, then we have spindle cell, spindle afferent. Afferent is towards the spinal cord. So, this will be this is a smaller fiber, spindle afferent, then spindle efferent. 1 beta. Okay. So, we will revise the whole stuff. Skeletal muscle, this is skeletal muscle, this is 1 alpha fiber which synapses onto the muscle cell. The huge spindle which is disproportionate, the spindle has got muscle cells on both sides, each muscle cell is fed by a 1 beta fiber on both sides. The muscle cell itself gives an output to the spinal cord through this spindle cell fiber. So, what we first thought, case 1. Case 1 is I have put a stimulus. Stimulus is stretch. So, that produces spindle afferent, that causes anterior cell activation, that causes skeletal muscle activation. So, spindle afferent results in ACN and then that results in muscle cell. So, clear about that. Case 2, we give spindle efferent. Now, what happens with spindle efferent is it causes spindle afferent activation. Why? Because there is stretch in the spindle. So, both the peripheries contract, spindle gets stretched, muscle is not stretched, spindle is stretched. What does this do? It causes ACN activation and that causes smooth muscle activation. So, that's the other way of doing it. Now, case 3, say ACN is activated, smooth muscle is activated, spindle afferent is also activated because spindle afferent is activated. So, it goes in this direction, it goes in this direction and it goes in this direction and you have plus. So, you have more contraction happening over here, but this does not cause it indefinitively. So, what will happen is a increase in this one causes further this in turn activates something else, the another reflex by which the contraction comes down. So, I think that did not be mentioned here. So, this is a beautiful loop which happens within the muscle. The nervous system is able to control muscle activity through this mechanism. Please note that each one of these 3 things are different. There is external stretch resulting in muscle contraction. There is efferent contraction, 1 beta contraction which causes activation which causes muscle contraction, and then there is alpha alpha stimulation which is causing further muscle loop. So, the fine tuning of muscle activity happens to a large extent in the spinal cord. Now, what are the implications? Implications are, there is I have not completed the loop. So, why does not this continue indefinitely? So, in the skeletal muscle design which I spoke about, the tendon which is that this one. So, when there is stretch in the tendon, this reduces the alpha activity. So, alpha activity which is coming to the neuron for the neuron. So, this is negative. So, when this is negative, so there is a balance between the spindle activity vis-a-vis tendon. This is from the Golgi tendon. So, Golgi tendon, spindle cancel out each other. So, you can't have indefinitive contraction happening. On the other hand, what is more interesting is there is this positive feedback which is there, and if you have just spindle afferent coming without any other activity, you notice that there is a feedback cycle of muscle contraction which is happening over here. I think I did not complete this. Yeah. So, that completes the loop. So, in effect what is happening is you have 2 or 3 different control mechanisms by which muscle cell length of the muscle cell is controlled. So, length of the muscle cell can be controlled by one alpha afferent, one beta, or by combinations of spindle cell and Golgi tendon activity. So, there are multiple things. Now, the sum of all these things, if we look at what actually happens with all of this is, there is something called as tone. So, tone is a resting state of contraction. What it essentially means is that no muscle cell starts at 0. So, there is always a baseline activation. So, if we plot time and state of activation, and 100% contraction and 0%, muscle cell is always at a particular this one. So, that level of activation is called as tone. So, when there is activity, there is increase in the activity to various extent based on the amount of stimulation, and there is also a provision that if you have say something happening on the opposite side, you can reduce the tone a bit, and you know that is responsible for a lot of functions which we go. So, this is how the idea that the monosynaptic reflex is a fundamental entity in controlling muscle activity is depicted. Let us revise. So, we start with stretch. Stretch is spindle afferent stretch. Spindle afferent stretch causes ACN activation, that in turn causes skeletal muscle activation. So, that contracts, and that contraction results in negative, and that in turn becomes negative, that in turn reduces negative. Now, if we look at the second, in which spindle afferent causes primary stretch of the spindle afferent, which in turn activates the alpha motor and in turn produces this one. But in this case, there is still persistent activation happening, because the activation is not from the spindle cell as such, it is from the muscle cells on the spindle fiber. Okay, what do I mean by that? We will redraw the spindle. So, this is what happens with the tendon reflex. Which results in decreased contraction followed by relaxation. Contraction because the length suddenly increases, stretch increases, and that produces muscle contraction. This is sort of resting state. On the other hand, when there is contraction in these two parts, this gets stimulated, muscle gets stimulated, muscle contracts, but the signal does not die out because the contraction is being manipulated over here. So, there is continued signal coming from this state. So, case 3, continued muscle contraction. And that is called tone. The state of continued contraction, which is there due to the muscle spindle and the muscle cells, spindle extrafusal and intrafusal fibers both, is responsible for the state of tone of the muscle. This sort of gives an idea of the richness of the network and the control mechanisms. Remember, this is just one of the most simple control mechanisms which are there in the human body. It is pretty conserved, and you can notice that, as I told you from the previous graph, tone is a fundamental property. The concept of tone I had already highlighted somewhere in the neural network discussion also, because there is a resting state of activation. It means the opposite, but it is a very beautiful statement, because it states that what we call rest already has some activity built into it, and what the nervous system does for control is to change the state of this activation. So, you can either decrease the activation or increase the activation to produce some activity, and that is a very fundamental property of all nervous system work. So, you always start at a baseline, including if you look at the resting membrane potential, which is there in all excitable cells, but this is one level above that. You would always find that the state function is at a higher threshold than what is necessary. It expends energy, but for whatever reason, maybe for control smoothness, or maybe maybe there is a control theory which explains the whole stuff, which I am not aware, but the state of activation is what is changed. So, what actually gets changed is the gain of the system. So, the gain of the system results in function, and that I think is a very profound statement when it comes to analyzing neuromuscular cell activity. The second thing which I want to say is the integral relationship between controller effector and sensor. So, the loop is there from the beginning. It is not that the loop is constructed, or what I mean to say is, there was never a time when the nervous system developed independent of the muscle. So, you can see that the muscle has a sensor embedded within itself, and it is that cell which is it is completely in tandem with the regular muscle cell, and the control is not just at that level. You have got even muscle cells present within that sensor. So, you have a effector connected to a sensor effector. So, sensor is also has got another effector in it. So, that is another beauty. So, you have, and these are not just one, there are literally millions of these cells, and that is the kind of richness which biological systems have in their basic control system. So, this is something very which is present at the heart of the whole system, and that is the reason I thought I should start my network analysis with the monosynaptic reflex. There are polysynaptic reflexes in which multiple other neurons come in between those two effectors and the sensor dynamics, and it gets modulated by various things. But I showed this in the demo at various muscles to say that it is universal. So, when we discuss, if you take a textbook of medicine physiology, you would find that the reflex is discussed in very bland terms as a method by which you diagnose, a method by which you you describe control within that, but more than it being an example of function, it is an example of the kind of hierarchical processing which happens. So, how does this reflex help in diagnosis? A brief thing. So, reflex, if there is increased 1 beta, it increase in tone, which basically indicates that there is a problem high in the cord or brain. So, if there is a cut between, so basically if it is decrease in 1 alpha, it indicates decreased tone and spinal segment problem. So, that is a very crude way of describing how the medical interpretation is done, but the theory remains the same. The theory remains the same whether it is discussed in medical terms or whether it is discussed in engineering terms. In engineering terms, what I would like to highlight is the use of gain as a method of control. I think that is the mechanism of servo controls, but the beauty of putting a sensor in the actuator with an actuator in the sensor, I think that is something very unique. So, that is, I think I should write that down. Sensor in actuator in sensor. So, this is something for you to think about, something which is very beautiful, and it is a very fundamental notion of the control systems within the brain and spinal cord. So, with this I think I will stop my discussions over here, and we will continue in the next class.